Lu Tran Hui Tang from Bugyeong University. And his topic, presentation topic is automatic bacterial nucleic acid extraction device using disposable and pressure-driven cartridge.
Thank you for letting me share our work at Biochip conference. Today I'm going to talk about an automatic bacterial nucleic acid extraction device using disposable and
Hello everyone. My name is Tang, and I'm from Bugyeong National University under the supervision of Professor Chung Chip. Thank you for letting me present our work at Biochip conference. Today I'm going to talk about an automatic bacterial nucleic acid extraction device that using disposable and pressure-driven cartridge. So what is nucleic acid extraction? Nucleic acid extraction is a method to isolate and purify nucleic acid, which is essential for further downstream applications such as medical testing and genetic sequencing. The conventional method for nucleic acid extraction can be classified into liquid phase extraction and solid phase extraction. And spin column-based nucleic acid extraction is one of the most widely used methods. It includes cell lysis, DNA binding, wash, and elution. And in between these steps, it requires multiple manual pivoting steps and centrifuge. So the advantage of this method is it's easy and safe. It can start with a wide range of starting material, and it is reproducible. However, it requires bulky equipment and also manually pivoting. And in general, it is not suitable for point-of-care testing. So it is important to automate the column-based extraction process by applying cartridge and microfluidics. But before going to my research, let's have a look at what is going on in the academia. So here are some notable work in point-of-care nucleic acid testing device. The first one is a paper machine that can perform nucleic acid extraction, isotomal amplification, and detection. The second one is a rotary microfluidics system that can perform nucleic acid purification, isotomal amplification, and detection based on lateral flow assay. And the final one is pop-up paper-based device that can perform extraction, isotomal amplification, and also detection. So, although recent significant advances have been made, there are still a lot of things that can be improved. So for the automation of sample in and answer out, there is still a need to eliminate the pivoting in between steps and streamline the lysis, amplification, and detection. So our goal is to automate the nucleic acid extraction process, and we want to make it easy to operate, which means no manual pivoting step. And we want to have a self-contained cartridge with reloaded reagents, so it can be suitable for point-of-care and eliminate contamination. So the concept of our device is we want to use pressurized air and to move the restore reagents through the microfluidic chips, and we utilize the silica membrane to capture the nucleic acid. So before the needles puncture the rubber cap, this is a closed
So
system,
we designed
and after the
3D
needle
printed
punctures the
disposable
rubber
cartridge
caps,
with
our
4
device
sets of
starts
tubes.
to operate.
Each step represents one step in
And
the cartridge
here is
extraction.
the design of our microfluidic chips, and
And the
it
part that
includes
contains the license
one
is separable.
PMMA at the top and one at the bottom, and in between there is
So the
there
3D
are
printed license
three BDMS layer with
consists of
silicon
compartmentalized
membrane.
reagents to light cells.
So we
And the
designed a
reagents are
3D printed
separated from each
disposable
other by
cartridge
a rubber stopper.
with 4 sets of tubes.
And we
Each set
use screw-type
represent one
plungers
step in the
to
cartridge
release the
extraction,
reagents and also to prevent
and the
backlash.
part that contains the lysis tube is
So
separable.
here is how the license tube operates.
So the 3D printed
So
lysis
first
tube
we inject
consists of
the
compartmentalized
sample into the
reagents
license tube.
to light cells. And the reagents are separated from each other by rubber stoppers.
And after we
And
draw
we
the
use
needle,
a screw-type plunger
rotate the
to release the
plunger to
reagents
release the
and
reagents.
also to prevent backlash. So here is how the lysis tube operates. So first we
And then we
inject the samples into the lysis tube. And after we draw the needles, we rotate the launchers to release the reagents.
So our cartridge and microfluidic chips are packaged into a 3D printed disposable box. So after the
And then we
license
shake
step,
the
the box
tube
can
to
be
mix
inserted
the
into a
solution.
machine to automate the nucleic acid purification process. And here's a clip to demonstrate how the machine works. So first we slide the
Our
box into
cartridge
the
and
machine.
microfluidic chips are packaged into a 3D printed disposable box. So after the lysis step, the box can be inserted into a machine to
Then we
automate
fix
the
the
nucleic
cartridge
acid
to the cartridge holder.
purification process. And here's a clip to demonstrate how the machine works. So first we slide the box
After
into the machine.
start button, the machine starts to operate. So as you can see, after
And then
the
we
needle
fix
punctures
the
the
cartridge
rubber cap,
to the cartridge holder.
liquid starts to move up into the outlet tube. So after 1 minute,
And
the
after
cartridge
pressing
moves
the
up
start button, the
and
machine
rotate to
starts
the
to
next
operate.
tube. So by repeating these steps, nucleic acid can be extracted automatically.
So as you can see, after the needle punctures the rubber caps,
So we want to
liquid
figure
starts
out
to move
how
up
much
in
pressure
the
we
outlet
should
tube.
put into these tubes and how long.
So
each
after
step
1
will
minute,
take. So we
the
did
cartridge move up
preliminary
and
study.
rotate to the next tube.
So first we load
So by
500 of
repeating this
microliter
step,
of
nucleic
ethanol
acid
into
can
the
be
inlet tube,
extracted
and we
automatically.
compress air into the inlet tube, or either we create vapor
So
pressure
we want
at the
to
outlet
figure
tube,
out how much
and then
pressure
we
we
measure the
should
height of the ethanol
put into this tube
move up in
and
the
how
outlet
long
tube
each
after the
step
needle
will take.
punctures the rubber cap.
So we did a preliminary study.
So
So first we load 500 of microliter of ethanol into the inlet tube, and we compress air into the inlet tube, or either we create vacuum pressure at the outlet tube, and then we measure the height of the ethanol move up in the outlet tube after the needle puncture the rubber cap. So in general, as the total amount of pressure increase, the time required for ethanol to flow from the inlet tube to the outlet tube decrease.
Perform the extraction and record the time
I
required
chose
for liquid and
150
gas to move from the
kVA
inlet to the
positive
outlet.
pressure
So
instead
on
of
average,
200
the
kVA
time to
because
process all the
200
liquid
kVA cause
and
leakage
air is
sometimes.
around 1
And I
minute
chose
30 seconds.
minus
And
kVA
the total
vacuum
machine
pressure
running
at the
time
outlet
is
to
below
have
4
the
minutes.
fastest operation time. So with
So
this
we
setting,
wonder whether the
I'm
3D
perform the
printing
cartridge
affects
extractions
the
and
immediate
record the time
qPCR
required for
results.
liquid and
So
air
we
to
did
move
a
from the inlet
comparison
to the
between
outlet.
the ED2 and
So
3D
on
printing.
average, the
when
time to
performing
process all the liquid and air is
using the
around
commercial
1
kit.
minute and 30 seconds,
And as you can
and the
see,
total machine running time is
the
below 4
3D
minutes.
printing tube didn't indicate qPCR results.
So we wonder whether the
And then we
3D
compare
printed tube affect
cartridge
the
extraction
inhibit
with,
the qPCR
the
result.
conventional method.
So we did a comparison between the
So,
EB tube and
as you
3D
can see
printed
here,
tube
DNA
when
concentration,
performing the
purities, and qPCR
lysis
results
step
are
using the
similar
commercial kit.
between the
And as
commercial
you can
kit
see,
and the cartridge extraction.
the 3D printed tube didn't inhibit the
And finally, we
qPCR result.
determine the limit of detection of
And then we
our
compare
device.
the cartridge
So,
extraction with
the limit of
conventional
detection of the
method.
cartridge extraction is 10 to
So
the power 3
as you can see here, the
CFU
DNA
per
concentration,
ml.
the
And
purities,
when
and
amplifying
qPCR
with
results are
granular
similar
scientific
between the commercial kit and the
which is
cartridge
comparable
extraction.
to the column-based extraction result.
And finally, we
And,
determine the
the
limit of
efficiency
detection
of the
of
standard curve is
our
91%,
device.
which is
So
within the acceptable range.
the limit of detection of the cartridge extractions is 10 to the power 3 CFU per ml. And when amplifying with grammar targeting RFPE genes, which is comparable to the column-based extraction results. And the efficiency of the standard curve is 91%, which is within the acceptable range. And the gel electrophoresis also confirmed that there is amplicon at 10 to the power 3. So in conclusion, we introduced a novel disposable pressure-driven cartridge with reloaded reagents. It is that automate the nucleic acid extraction process. It is rapid. So the lysis step is around 30 minutes, and the cartridge extraction is 4 minutes to extract E. coli DNA. And our system is a closed system, so it prevents contaminations. And the result is we have comparable limit of detection to conventional methods. For future work, we want to automate the lysis process. We also want to handle more volume of samples, and we want to integrate the amplifications and detection modules to our device so that we can achieve sample in, answer out at point of care detections. So thank you everyone for listening, and thank you Professor Shin and everyone at our lab for support.
Wonder about the utilization of your
Okay, thank you for your nice talk. If you have any question, feel free to ask him.
from your system.
If there is nobody, I will ask one question. I just have a wonder about the utilization of your extracted DNA. What, how can you use the DNA which we extracted from your system?
So your question is how can I utilize the DNA? At first we develop a system to extract the DNA so that we can use it for many other applications. But at first we perform qPCR to show that the DNA that extract from our device can be performed qPCR. And in the future we want to integrate that with amplification and detection so
오늘
we have
교육실 내용은
our device can perform all these steps together.
포스트라는 주제입니다.
Okay, thank you for